Our first speaker in this session is Dr. Saber Hussein. Dr. Hussein is a senior scientist in the Human Effectiveness Directorate of the Air Force Research Laboratory of the United States, located in Wright-Patterson Air Force Base in Ohio. Dr. Hussein is a lead scientist. He focuses in his research on the development, direction, and performance of state-of-the-art research in the emerging areas of nano biotechnology. Neurotoxicity, of course, or nanotoxicity, of course, I'm sorry, not neuro, I'm just bringing my specialists to you here. Dr. Hussein involved in many research work on nanomaterial interactions with living systems, including in vitro and in vivo studies, intracellular fate of those, and uptake, of course, of those nanoparticles or nanomaterials, and their potential toxicity, which is an important subject for all the people who are working on this field of nanotechnology and nanoparticles. He is looking for novel nanobiotechnology and nanotoxicology model development, and thus in order to enable safe implementation of nanomaterials and nano products. Please welcome Dr. Hussein. Hope that we can have 15, 17 minutes for each one to present, then we can end up with some time for discussion.
Okay, thank you. So my time starts now, right? At the outset, I would like to thank Dr. Saleh Suleiman and organizing members for inviting me to give a talk here. It's a very informative meeting. Really, we enjoyed the immense hospitality that you all provided and the friendly environment. Really, it's a great meeting, and I'm really thankful for the organizers for giving this opportunity. Today, what I'm going to talk about, evaluation of engineered nanomaterials and the toxicity. In particular, I would focus how this area is different from traditional toxicology and how this is linked to some of the physicochemical properties of nanomaterials. So this is like an outline of my presentation that you can see. I would talk about a brief intro on the nano and some of the key unique physicochemical properties of engineered nanomaterials. And then I would talk about some of the risks associated with these unique materials and what are the challenges for toxicologists to come up some toxicological evaluations. And then I would talk about what are the opportunities does this nanotoxicology present in future. To give a brief introduction that, you probably all know about the nanotechnology, and the nanotechnology, the fundamental aspect of nanotechnology is the engineered nanomaterials, which deals with the nanoscale from 1 to 100 nanometer. If you compare to the size here, it is relatively in this area at the nanoscale, and the 1 nanometer is approximately 1/1000th of a meter. So you can get some kind of a sense what is the nanoscale compared to the bulk material. So which is really, we are talking about at the level where the scientist can manipulate these materials at the atomic level, and then it won't follow the classical physics, but it follow the quantum physics, and that really enables scientists for manipulating for variety of applications. So some of the applications I will talk about in the next slide, but some of the key physicochemical properties that how they manipulate, and you can see some of them are here. The size, shape, charge, and coating, but there are other parameters that can be manipulated at the atomic scale level that makes this technology really very unique. In the next slides, there are some of the impressive unique properties. One is the nano size. And when any material or the matter, when it goes to the nano size and its surface area significantly increases, and obviously when surface area increases, its reactivity increases, and it keeps its atoms very available in the matter, more available, and make it susceptible when it comes to biological interaction. So the size and the surface area, and when the surface area increases, its catalytic activity also changes, and more importantly, their optical properties. So how they interact with the light, and one of the best example is a gold nanoparticles, how it can be tunable with regard to its size. When you change the size, its color changes significantly. And then once the color changes, you can see its peaks, and it shifts its spectra. And the same material, when you change to the size, it displays variety of color, and that's an excellent property for developing therapeutics or diagnostic tools. Therefore, it has variety of application, biomedical applications. As you heard, one of the applications for the cancer treatment in the opening ceremony lecture, Professor El Sayed, he gave excellent talk, how the gold particles can be manipulated because of their photothermal properties for therapeutic application, in particularly for the cancer treatment. And besides that, they can also manipulate their aspect ratio. I don't want to go in details about the material aspect because of the time, but the point here, the take-home message, that these materials at the nanoscale level, you can manipulate their atoms by atoms, and that will have a key application, not only for diagnostic, but various applications. Some of the applications that I have listed, the consumer applications, medical applications, and the industrial applications. So these are some of the examples I gave you, but one way or other way, all the materials that we are using now in your hands, the cell phones and all these electronic tools, even consumer products, and they do contain these engineered nanomaterials. So while there is a beneficial properties or the unique properties, impressive properties, but there are certain challenges. And what we worried about as a toxicologist, whether they have any potential risk associated to produce some of the health implications, and what are the toxicological ramifications based on their unique characteristic features like size, surface area, shape, and optical properties, magnetic properties. What I'm looking, when you manipulate these properties, so that will change any toxicity and biocompatibility, and also these materials, while you are dealing and developing these materials, there is a great concern about impact of these materials in the environment and also on the human health. There are variety of pathways how we expose, through inhalation or through skin, and some of the medical application, if they are in the consumer products, it's also likely to go through the ingestion. So there are variety of pathways that we expose. The bottom line here is, because of their unique properties, it is really critical for the technology to be safe. We have to evaluate the toxicological implications. So one of the things that I try to look at, what this technology, how they produce the toxicity. And I will give you some examples what we are doing in our laboratory, and I will talk about how these parameters like size, the surface area, some of the physicochemical properties are key and important while we are looking some of the toxicity. And here This is a silver nanoparticles, and we know that the silver had become very popular now because of its antimicrobial properties and its optical properties. It has a lot of applications now. And if you look at the left side, we have to characterize these materials, and that is what it makes very unique and different from traditional toxicology. And looking at that electronic microscopy, the size of the materials we have to evaluate when we get the materials, it's at 20, 40, and 80 nanometer. And as a classical toxicologist, you put it in the media, and then once you put it in the physiological media, and obviously because of their intrinsic nature, they kind of tend to aggregate and agglomerate. And that is one of the challenges that the toxicologists they are facing. Once they aggregate, you can see like the primary particle size 20, 40, and 50, but once they are in the media, because of variety of composition in the physiological media, they kind of aggregate, and then you will see the aggregation, they completely change the size of the particles. For example, once they aggregate, the 40 nanometer tend to aggregate, and that becomes little lower size of the agglomerated in the media. So the point here is then once you expose to the cells, and we have to make sure that they are really translocated in the cells. And one of the key features is all these materials, they aggregate and they are taken up by the cells. And most of the cells, they are taken up into the vacuoles, in the endosomes. And that is another property of these metal-based nanoparticles. They are embedded in the endosomes. And once they are, and what I'm trying to show here, that based on the size, and a lot of people in the literature, they have reported that the toxicity of these materials based on the size. However, we have to be careful that the size is not the primary size what we get, but the size once they are in the physiological environment. So that is what we have shown. The 40 nanometer, which shown to be more less in size, once they agglomerated, they produce the more toxicity here. They also produce some reactive oxygen species. And some of the key genes that when we evaluate, some of the genes involving in the oxidative stress also had been induced significantly. So again, the silver nanoparticles exhibited its toxicity based on the size, but also one of the complexity that we found to characterize these materials once they are in the cells, the extent of agglomeration and the aggregation in the media or in the physiological environment determine their potential effect. The same thing here, the silver. The another thing that if we take the silver at the low dose, which not necessarily lethal to the cells, whether they produce any signaling pathway. To make a point short here due to lack of time, and we always characterize them to make sure what their size is in the agglomeration in the media. And then we looked at their uptake by electron microscopy to look at their fate and kinetics of these materials. And then when we looked at that lower dose, what it did really, it impacted the ERK pathway. So the signaling pathway at the 5 microgram, even just for the acute exposure at the 24 hours, and you don't see the cell death, but it has some great implication on the signaling pathway because it is inducing reactive oxygen species. So, and also we try to look at some of the phosphorylation, which is a key event to trigger the signaling pathway. And this is one of the universal pathway for the signaling that cover most of the cell function, proliferation, and so on and so forth. So the silver, even though it is not lethal at the concentration we have tested, but it has some impact on the signaling. And the point here is that if we continue to expose to these materials, even at the lower dosages, the long-term implication of these materials would impact the cell behavior and the cell function. And then it is again, even the gold. So gold is inert particles, and gold had, everybody known, which is non-toxic. But point here, what I'm trying to show, that when they become at the nano size, and you can see that there is some toxicity, and I don't want to go in detail here, but the gold also has two kinds of materials. One is the spheres, another is the rods. So the rods, It depends on the aspect ratio. You can make it like 10, 20, 50. So change the aspect ratio. Interestingly, what we found when you expose the cells with the shapes and the rods, which is different aspect ratio, have tendency to show some sign of toxicity. And when you compare to the material aspect, really in order to make the rods, they have to make some detergents in order to grow them, and it is completely the material side. But the rods, they have different applications because of their catalytic and the surface area. They have a huge biomedical application. The point here is, if we don't look at the safety and the progress of these applications into various devices could impact, because we have to evaluate their health implication before these materials designed so that we can generate the safe environment. And the other aspect that we have several publications. If you are interested, you can look at them on the literature. Due to lack of time, I'm not going to show all of them, but we really did comprehensive studies, the toxicity of these materials based on the size, based on the charge, based on the shape, and based on the surface chemistry as well. And that is the beauty of nanotechnology, how you manipulate, how you functionalize these materials. And we have to look at, once you functionalize their surface chemistry and all those parameters, in one way or other way, they have some kind of implication. I'm not saying every material is toxic. Some of them are toxic. Then we need to convey to the material scientists so that they can resynthesize in a manner that are more biocompatible and safe. And some of those you must have heard about the carbon nanotubes, and they also shown some of the sign of toxicity reported by other people. It actually published in Nature by European group, that they have shown some of the toxicity quite relevant to the asbestos. Carbon nanotubes, they are longer in size, but one dimension is the nano size. And then also they have shown that they injected to the mice for 2 weeks, as a occupational exposure for 6 and 8 hours of exposure, and they found out at the molecular level some of the proteins that induce, they likely to suppress the immune system of the mice. So there are some indications for the carbon nanotubes as well as the metal-based particles that they produce some toxicity. And some of the challenges, just quickly to go, and the agglomeration that we studied, copper and the silicon as well. And most of the complexity in the cells depends how these particles aggregate each other in the solution. And then we also really looked at some of the behavior of these particles in the various in the water and the alveoli and the lysosomal fluid, to make sure how they interact with these fluids once they accumulated into the body. And they have a variety of sign of toxicity when you introduce these materials in the variety of media or in the physiological environment. And one of the thing also, the dissolution. So these materials, they have a tendency to leak the ions, and one of the most notorious is the silver and the copper, and because they have some oxidizing nature, and once they are in the media, they slowly leaked out. And that's one of the issues that most of the people that were using, some companies, they are using silver nanoparticles as an antimicrobial for a lot of clothing, that they are coating the fabric with the silver nanoparticles, and that has become one of the issues in the washing machines, because when they wash these clothes and they are leaking the ions and they are contaminating. So one of the issues here, it is not because of the nano and because of the dissolution constant that's slowly releasing the ions. And we did comprehensive study, and we actually published in the Nanotoxicology about a year ago. Then you can see the comprehensive analysis of ionic leakage from these. And the second aspect, the challenge, is the fractal dimension. So once it is in the media, the way they change their aggregation, so it is called fractal dimension in the physics language. If we calculate the fractal dimension and relate to their bioeffects and the toxicity, or even in general the bio interaction, there has a lot of significant application as well to develop some of the sensors based on how they aggregate in the cells. And the dosimetry is another issue, because in a traditional toxicology, they really have based on the mass, but in the nanotoxicology, we really have to look at based on the surface area. And some of these that you can see how we do characterization, looking at the detail about their size by dynamic light scattering, and some of the advanced techniques, and all these things in the cell. So there are two pieces. The one is left side, their physical properties, and the another is their interaction with the cell. So that is where I'm trying to indicate that the physicochemical characters and the characterization is a key point. So how can we improve? And some of those, then we have some kind of solutions for that you can see, and I would be happy to help for calculating the dosimetry. And some of these lists that you can see, we can go step by step for target dose, dosimetric, and methodology, and the dose characterization. And also we are trying to enhance these in vitro models in a manner very close to the in vivo organ system. So the best way to look at the 3D models and 3D co-culture model to simulate the in vivo environment. And this is one of the review article in the Toxicological Sciences that you can see the crossroads of nanotox, past achievements and the current challenges. This is one of the nice comprehensive article that describe what's happening for the past decade about the nanotoxicology. The point here is, I would just like to conclude here, what are the future opportunities? And I have shown some of the nanotoxicology concept, how the toxicology of engineered nanomaterials depends upon their physicochemical properties, which is clear in the cells. And then the other important thing is, in order to evaluate the nanotoxicology, it relies on its chemistry, so we have to work with the chemist, and also it relies on the engineers for the material development, but it also relies on the regulatory agencies, because end of the day, once we do, and we have to develop the standards through the regulatory agencies. I don't know, some of the picture disappeared here, but then also there is a huge application. Once we know how these engineered materials interface with the biological system, if we understand the complex interaction in the cells based on these unique features, there are so many other ways that we can use these materials in the nanomedicine, and that is one of the novel areas that people are working. So the point here I would leave the nanotoxicology bringing to the table the chemists, the material scientists, and the regulatory agencies, and the medical people. So, and for all of them, the safety is the first. If we evaluate the safety of these materials, and all the material development and application would go very smoothly. So with that, I would like to thank my team. If you have any questions, I would be happy to take it. Thank you.
We'll have the question at the end of the session, but we thank you very much for stick to the time. And as you have seen from the talk of Dr. Hussein, health and environmental impacts of new materials must be studied, and the risk assessment must be evaluated way before we think of adopting those kind of materials in order to save public and the environment from getting affected by such new products. Our second presenter is Professor Dr. Jeffrey Jenkins, an old friend of mine as well. Dr. Jenkins has received his PhD from Michigan State University in 1981, and he is a part of the team that developed a new class of insecticides called ivermectins group. I'm afraid for the honeybees. Yes. But let me just, to save some time, because Professor Jenkins actually has lots of work in the area of environmental toxicology and environmental impact of chemicals. And also he has lots of international efforts, primarily on the sub-Saharan Africa, North Africa, and the Middle East. Together we joined in many workshops regarding the harmonization of MRL of pesticides, as well as the regulation of pesticide as well. Dr. Jenkins will also be one of our group who will organize the next or the second round of the risk assessment training course here in Bibliotheca Alexandrina next August, hopefully. which will be followed also by a workshop on food security in collaboration with the United States Department of Agriculture and the Foreign Agriculture Services of the United States and other cooperate in this regard. So please welcome Dr. Jeffrey Jenkins.
Well, good morning. I'm glad to be back in Alexandria again. Here we go, because I have too many slides for the amount of time that I've been allotted. So we're going to talk today about during this conference, there's been a number of discussions of what is sustainability and the concern about feeding the world. Thank you. So I'm not going to spend a lot of time on this. This is just for introduction, but this is one resource, the World Resource Institute, and sustainability is needed to close the food gap, reduce agriculture's impact on climate, ecosystems, and water, and that's going to be the major focus of my discussion today, and then also the economic and social considerations. A second definition of sustainability, much of it coming out of a UN General Assembly document, but it gets again back to the core. Sustainability is lasting success requires an integrated approach, and again we have production, farm profitability, quality of life, and then finally stewardship of natural resources. And then another statement that they made in this document is that sustainability requires recognizing and acting upon productivity, economic, social, and environmental goals as a simultaneous set of system attributes. And so again, another focus of what I want to talk about today is this: how do we define sustainability in terms of environmental impact, and do we need to use a systems approach to adequately evaluate whether a sustainable practice is meeting or whether an agricultural practice is meeting our sustainability goals. And again, this is from the United Nations Environmental Program, and I don't expect you to be able to read anything on this graph, but the statement says, the framework attempts to reflect key components of a complex, multidimensional, spatial and temporal chain of cause and effect that characterizes interactions between society and the environment. And again, sustainability goals, how do our food production goals meet sustainability objectives when it comes to environmental protection? And this is going to require, just from looking at this diagram and not really knowing what it says, a systems approach. And yet another diagram that is focused specifically on agriculture. And you can see that there is a multitude of boxes up there, and I don't expect you to take them all in. But at the bottom, you've got feasibility of production practices matched up with environmental relevance. And how do we meet both goals? How do we meet production goals and meet our environmental protection goals? So I'm going to spend most of the time with a case study out of the Pacific Northwest to kind of demonstrate one approach to how you might meet both these goals, the food security and production goals, and balance that with the environmental protection goals. And so in the Northwest, salmon, some on its species, are an icon of the Pacific Northwest. They're culturally an icon, and they're socially an icon, and they're endangered. And so there's a major effort to protect these fish species and at the same time continue the agricultural practices that are going on in that part of the world. And so this is a map of farm resources in the U.S. And if I can figure out how this works, I guess it's this way. There it is. And so this part of the U.S. is the area that we're concerned with. California and the Pacific Northwest, a major agricultural production region in the United States. California has the 8th largest economy in the world, and it's just one state in the United States. And let's layer over the top of that the distribution of endangered and threatened salmon and steelhead species. So these 5 species break down into 28 evolutionary significant units that occupy that this is their distribution and habitat. And this overlays a very important and expansive agricultural production region of the United States. So how do we continue, or how do we try to meet sustainability goals on this large of a scale and at the same time protect these fish species? What do we need to know? How do we go about understanding what is the environmental impact of agriculture on these species? So it starts with, what do we know about them? What is their occurrence? And I just showed you their distribution and habitat range. Are they susceptible to pesticides that are used in agriculture, for example? And what is their behavior that might result in exposure? And then you balance that with where the pesticides are used, and that translates into opportunities for pesticide exposure. And where those two overlap, where the pesticides are used and where the species occur, is where the risk lies. And so how do we evaluate the risk of agricultural practices to some monid species? Well, this is a diagram that indicates if you put chemicals into the environment, they get distributed in the environment based on their physical chemical properties and based on other and based on environmental conditions, which I'll get to. That results in environmental fate, which in turn results in potential for exposure. And based on bioavailability, there is exposure to the organism. And then based on toxicokinetics and toxicodynamics, there's internal exposure, and then there's effects. And what we really need to do is balance our understanding of the fate of the chemical in the environment with the toxicokinetics and toxicodynamics in the organism. And so do we know on the appropriate time step where the chemicals are in the environment and where the fish are in the environment, where they intersect, and what time step is important? And so if we think about salmon biology, we also have to think about the various life stages. Which of these life stages are more susceptible to chemicals than others? And as you know, salmon are anadromous species. They spend, once they hatch, they spend a year or more in fresh water, move out to sea for 2 to 3 years, and then come back to spawn. And so they run the gauntlet from where they hatched, going out to sea through all of this agricultural land, and then when they come back to spawn, they once again have to travel through all of this agricultural land. And where are the opportunities for exposure, and what time step is adequate to understand that exposure? And then there's also not only the fish, but the fish food and the entire food web. And in order to protect these salmon, you also have to protect the food web. And so now we have many, many aquatic organisms that also may be impacted by chemicals used in agriculture, and how do we understand the interaction between where those chemicals are in the environment and impacts on the entire food web. So we've got in the Northwest, we've got pesticide occurrence based on where these chemicals are used and site-specific conditions, and then the salmon biology, life history, and habitat preference, and that results in exposure. So how do we understand that? Well, one way to understand it is go out and collect a lot of water samples and analyze those water samples and find out what's in the water, and then make some inferences based on sampling in the environment, what the patterns of exposure are. So we're going to look at, as an example, some salmoniid critical habitat in Oregon. And this is the state of Oregon in the Pacific Northwest. It's sandwiched between California and Washington. And the Willamette Basin is a major agricultural area. And the Willamette Basin is at what's called the Hydrologic Unit Code 4. That's a relatively large unit code. And then within that unit code, there are 12 subbasins that are at Hydrologic Unit Code 8. And the agriculture is very diverse in this part of the U.S. 170 crop varieties, and that results in more than 500 pesticide active ingredients being registered in Oregon because of the crop diversity. So how do we understand all of those pesticides used on all of those crops? How do we understand how that might impact exposure to salmonids? And so if we break that down into just a smaller Huck 8 watershed, this is one that we've been working in, and then we go down even farther to a Huck 12 watershed, and we've done extensive work in this watershed trying to understand the relationship between chemicals used in agriculture and how that might result in exposure to salmon. And so we looked at this one watershed, the Zollner Creek. It's one of 388 watersheds like it in the Willamette Basin. And we're studying just one of them. And we chose it because there's been frequent detections of pesticides in this watershed, and it's been used by conservation biologists. This small watershed, because it's been monitored so extensively, has been used by conservation biologists. The numbers that have come out of this watershed are used to imply that these concentrations are found throughout the Willamette Valley and Oregon habitat for salmon. So can we use monitoring data to adequately understand the potential exposure to salmon? Probably not. And so it's going to require both monitoring and models. And this is a statement from a National Research Council document in 2007 that says the spatial and temporal scales on which environmental controls and environmental quality are linked generally do not allow for an observational approach. That's the monitoring. To understand the links between economic activity and environmental quality, these linkages are made by modeling. And so this is the systems approach that I'm going to demonstrate. Models and monitoring are necessary to reduce uncertainty and to adequately frame the relationship between production practices, water quality, and to inform best management practices. And so we took this watershed, the Zollner Creek watershed has 600 fields and 40 crops grown in this small area that's about 3 miles by 5 miles. And this is some of the data over a 20-year period, some of the data for a few pesticides. And we chose atrazine as an example chemical to demonstrate whether a modeling approach might help us better understand the relationship between agricultural practices and surface water contamination. And so here's the data. Here's the data from 1 year of monitoring. And what we have here, if I can go back, is these are instantaneous grab samples collected seasonally at 2-week intervals. And there's high uncertainty, and this leads to very conservative assumptions about exposure. So here's the data, and here we have concentration of atrazine, and here we have Julian date for the year 2011. And you can see these are the detections of atrazine at the outlet of that watershed. What does this tell us about exposure to salmon? Well, we know that atrazine is there. We know the concentrations, it's there. But is this data adequate for us to understand the impact on salmon? And so we took this watershed, approximately 15 square miles, and you can see the stream network, and it's got a gauge at the mouth here, where we know what the stream condition is at the gauge, and so we've got a flow gauge. And so we used a model called the Soil and Water Conservation, or the Soil and Water Assessment Tool, And this is a watershed scale model. It's been developed over 40 years by USDA Agricultural Research Service scientists. And it's used to evaluate the impact of land management practices on hydrology, soil erosion, sediment transport, nutrient fade, and pesticide fade. It's a physically based process model that operates on a daily time step. And this is an important point, that it's got a fairly short time step at which you can evaluate or predict stream concentrations, which is not really practical if you're out just collecting water samples. There are very, very few studies around the world where someone goes out and collects a water sample every day for 360 year-round on a continuous basis. It's just not technically and it costs too much. It's not technically feasible, it's not economically feasible. And so here is the model in a nutshell, and again, I don't expect you to be able to read this, but the point is that there's about 750 input variables into this model. And it's got a weather component, a land management component, which also includes a crop growth model. There's a pesticide component, a nutrient component, an erosion component, a hydrology component, and then these arrows that point down into the stream, and then there's all the processes that occur in stream. And so there's an in-stream component. And so we took this model and used it in Zolna Creek. And part of the parameterization involves GIS data layers. And so we've got the land use, land cover data layer, the hydrology data layer, the soil data layer and the elevation and slope data layers all go into this model. And we use this model based on those input parameters to do an ecohydrology evaluation. And so you take a mathematical model that's designed to simulate the real world. And so when did events start in the real world? Well, millions of years ago. So how do you set up a model so that when you want to do your study, it's been running for a while, so that it's warmed up, is what the modelers call. And so you use weather input data, and you run the model for a number of years to warm it up. And so we ran this model for 5 years, from 2000 to 2005, with the weather input data for those years. And so that sets up the water balance within this watershed. And then we used another 2 years to optimize the model. And that's comparing our predictions of how much water moved out of this watershed at the gauging station with the actual gauging station data. And we use a number of statistics to evaluate this, something called a Nash subcliffe efficiency, a percent bias, and then a root mean square to evaluate the data. And that's a long and involved process. I'm not going to show it here. This is an example of the statistics we use, and then there's some performance statistics that tell us whether it's good or very good or unsatisfactory. Then the next thing that we did is we attempted to estimate the atrazine use patterns. So we went to the information about what chemicals are allowed to be used in Oregon that contain atrazine and what crops they can be used on in Oregon. And that's what's called the registered uses. And so we found that these were the potential uses. So out of those 600 fields, all the fields indicated here could receive atrazine, legally receive atrazine. But then we went to the local, we sought local knowledge from the crop advisors, and they said, well, actually, in 2010, atrazine was used on 10 sweet corn fields and 26 Christmas tree fields.
2 minutes, Jeff.
How much?
2 minutes.
Okay. And in 2011, it was used on these fields. So you can see that that's a much smaller set of fields. And so we use a probabilistic evaluation of application patterns. We established boundary conditions for the applications. We use spatial and temporal randomization using Monte Carlo methods. We did 500 simulations, and this is what we got. And so those points that I showed you before, we've now filled in on a daily basis our estimates of what we think the atrazine concentrations were based on the modeling effort. And they line up fairly closely, and particularly, not the green line, that's the absolute maximum simulated. But if you look at the dark line and then the red line that you can hardly see around it because the 95% confidence interval is pretty tight, you can see that we did a pretty good job of simulating atrazine concentrations. And so now we can take this validated model, and we have data now, simulated data on a daily time step, which is much more relevant to evaluating the potential impact on the endangered species. And you can see again, this is the distribution of those fields. And so what does this tell us? Modeling and risk assessments, models and geospatial methods can reduce uncertainty in the spatial and temporal relationship between pesticide use practices and receptor exposures. Models give context to monitoring data, and iteratively, monitoring data can be used to evaluate models. Mechanistically based modeling approaches are transferable. So we've done this work in this small watershed in Oregon, and based on our understanding through these modeling efforts, we think we can extrapolate that to the hundreds of other watersheds this size in the Willamette Valley. And models facilitate a probabilistic assessment, so we can look at exceedance probabilities, and models allow the evaluation of what-if scenarios, so we can go back in and say, well, what if twice the fields were treated with atrazine, et cetera. And so, for the Salmonid species regional assessment, there are almost 8,000 HUC-12 watersheds, and within those, within the 222 HUC-8 watersheds, and over that entire range along the west coast of the United States. And it's going to take a systems approach, it's going to take a modeling approach to adequately evaluate all those. It's not going to happen with simply going out and monitoring and looking for what's in the water and trying to make some sort of prediction of how that might harm salmon. So I think I'll stop there. And finally, down here, 300 crops, hundreds of pesticides. What about mixtures? And this approach also could be applied to the study of honeybees. So I'll stop there.
Thank you, Jeff. Thank you very much. You see from the approach that being taken by the modeling made by colleagues at Oregon State, that you can use the data available to rethink of what type of pesticide you use and reduce them to make sure that you meet all those requirements for preventing health and environment from getting exposed to pesticides. Our next speaker is Dr. Taysir Suleiman. Someone asking me outside if I have a relation with Dr. Taysir, but I told them the name is very common here in this area of the world. Dr. Taysir hold a PhD degree in information system from the Faculty of Computer and Information Science, Ain Shams University in 2004. So most of her work, I think, will cover that part of using computer and information in analyzing data and come up with conclusion. So please, for the sake of time saving, receive Dr. Taysir. Please, Dr. Taysir.
Okay. Good morning, everyone, and thank you very much for inviting me for this talk. I'm gonna talk about big data and environmental sustainability for biodiversity issues, and the focus on the biodialogue. Actually, I'm focusing on the informatics part within the environmental issues and the biodiversity issues as well. My outline down here is environmental risks on biodiversity and the biodialogue project where I work right now with a couple of universities will be discussed now, and the research opportunities for the road ahead, which is the theme of here of BioVision Alexandria. Actually, on the Global Economy Forum, which is just came out on 2016, is telling us like what are the global risks that we are going to suffer, and we need to concentrate on such global risks for our globe. And one of them was the major biodiversity and loss and ecosystem collapse. And actually, this also they like make a whole circles out of here from the water loss and water crisis we're suffering all around in specific parts of the regions, and the food crisis and their effect on the habitat loss of the biodiversity. Actually, another thing is the climate change that we see here in our world, and we can see it every year. Every part of the world is changing from the climate change. What is the effect of those climate change on the biodiversity, and what's the effect on our species as well, and which has like a direct or indirect effect on our human life and on our health as well. This is like the project we are working together from. It's funded from the DAD. And this project is actually called BioDialogue, which is a biodiversity intercultural communication. The project we are working on is consists from 4 main partners. The main partner in Germany is the Friedrich Schiller University, at Jena, Germany. And the main partner here in Egypt is Asyut University, coming from the Faculty of Computers and Information, and also at Ain Shams University and Faculty of Computers and Information, and presented by Professor Ibrahim Fathi Mawad, and Safakis University at Tunisia. And one of the interesting parts is that we came all together from the 4 main partners to have like main objectives for the biodiversity issues and how it is affected by our climate change for the 4 main countries. Like what we need to do was to mainly conserve our data for biodiversity and species from loss, and to need to have like some sort of databases for each country, and to spread out the knowledge of the biodiversity itself and the biodiversity informatics. This project actually, we called it like we have a theme for it. It's biodiversity without boundaries. And this is our main project director, who is the Professor Birgitta Kong-Rice, and Dr. Said, who is the project coordinator. And this is the 3 of us. And actually we had 2 phases. The phase 1, where we had is like our meetings together and the summer trainings for us. Mainly, like I know that most of you, or the audiences and also as well the speakers, they are talking about the nanotechnology and the toxicology, and also as well the environmental impact and all the species of the salmonids, as Professor Jeffrey was speaking about. And those workshops and the outcomes, we are mainly concerned with scientific data management. We're coming from a part that's like we have a computer science background, so the main workshops was training us on what scientific data management was and what is biodiversity as well, and to spread out the biodiversity informatics issues here in Egypt and in Tunisia as well. These are pictures from our Asyut workshop that we did on last November. And the most interesting parts that we went in Germany, it was one experiment, a Jena experiment, one of the most huge ones in Germany, that they were getting all information about different kinds of species down there, and they get the information of every plant there, and we went for a field trip to get the data, and to also conserve those data on different kinds of databases. And also, this is a botanic garden there in Germany, in Jena, where they have different kinds of rare plants, and we see it and how we can conserve such data. We are running right now our next phase, which is the second phase down there, where we have like a scientist and a student exchange, and also we are running experiments that I will show it to them right now. Since we have from the environment the impact on the human health, whether it is the individual and then the society and then the whole environment, we suggest like we are a group of computer scientists where some other scientists from the Environmental Institute at Ain Shams University. Like this part we can do, and these tasks we can do for the research opportunities for the road ahead. What can we do to save our environment? How can we do this? Through conserving our species. So we like first like we can develop instruments for engineered nanomaterials in the air. That's one issue from a technological point of view. We can develop tools to evaluate the toxicity. What is the level of toxicity right here? Evaluating the impact also as well of risk hazard and the impact on the environment, and then through these tools, how can this can affect the human health. So what we are doing right now, as a scientist, you can just go to a computer scientist, scientist where I mean like somebody from biology background, and he gets the data, and then he just give it to me, whether it's like a spreadsheet, where is it a sensor data from the GIS domain, whether it is the satellite data down here, like this one, and we had the experiments on the field, and the spatial distribution all over the countries where these species are mostly like the hotspot of species or the hotspot of the habitats, that they are disappearing right now, or there are just a few of them due to the climate change. Other issues we're suffering from is like the field inventories. Where can we get these from? And what we can get also from collecting the data is a very new field that we are working right now, is some people called citizen people or citizen data, where they just go ahead and they help the scientists to collect the data from different kinds of fields, whether they are images of the plants or just they have like different kinds of species or birds where they collect the data and they just send us through the images. So the diversity of the data itself came up with other research problems that we are suffering from right now, and we're trying to work with the other scientists to solve these problems. Such problems as like data extraction. I need to extract the data from different kinds of sources. Such problems will be when we give me the data, I need a standard to standardize all the data itself. For example, when a scientist by himself put the data, like for instance the temperature, he will just write temp. Other scientists will write T, other scientists will write the temperature by itself. For us, we need to standardize all of this data, and we are right now writing a research paper on how to extract such data from scientists' point of view. Another thing that we are suffering from in this field is the big data issue. And the big data issue that now on the fields right now, like everyone is saying, what's the big data? Where is the big data? Everyone, we are using every day on our everyday basis, like the Facebook, it's a very huge amount of data, that's big data. Mobile devices, like tracking all the objects all the time. Flickr, YouTube, this is generating a huge amount of data that we need also different kinds of equipments and high-performance computing that we need to analyze and discover and visualize all this data. So the term when it comes to big data, what does big data will serve the environmental part? The environmental science down here is a big environmental data. We can just get the data like chemistry measurements and the spatial distribution and this unstructured data that I need to confirm and that I need to convert to structured forms. Plus, these kinds of big environmental data that just came out like in a research paper in 2015, and it's telling me, well, you got observations, you get the water, water issues here, where the water goes, the level of the water goes down or up, and besides the biodata and the sediment as well. So let's now get into a platform that we can solve through the big data. that we can provide you services, like the scientist will just give me his own data, and my service will tell him, okay, this data is analyzed, and whatever kind of data you give me, you don't worry about it, and that's it. We can visualize it for you and see where is the distribution part. Another issue that came out of this project, that we have a group of students right now where they are generating like a tool. It's called a generic platform for citizen data. Whoever the citizen data comes and just go into the field, gets you a couple of images, and the thing is that when he gets a couple of images, he will upload his image, and you can, as a scientist or even a general user, just rank this image or recommend this image, that this image is very useful for whoever people who's working in the botany department, whoever people who's working with plants department and so on. And there are some tools right now that are trying to help the citizen data. One of them is called EpiCollect, that we are trying to do somewhat similar through smartphone data collection. And also the other one is called PlantNet, and the PlantNet is also like a software for automatic plant identification. So these kinds of apps that we are trying to develop is just to serve the scientists and even generic people, whoever are interested in such conserving the data. And this is our main target, and also not only conserving the data, but finding like similar phylogenetic problems and similar evolutionary data through the phylogeny that we can develop from. Now also what we are doing is that we're doing the data conservation through a biomap, through our lovely and wonderful botanic garden here in Egypt, like the one in Aswan, and the other one like the Sinai butterflies, and different kinds of databases through this. So the research directions that we are trying to solve is like we are trying to develop and link the diversity information part. We like also to enlarge our network of research network through the biodiversity informatics throughout different kinds of different countries. We are also finding the relation between metabolomes and transcriptomes and the proteomes of the species, and beside that, different kinds of learning standards like the EML, and last points where we were talking about the data integration part and the citizen data management, and also scientific data management, which has brought to us a lot of research doing there, like data fusion. Thank you. I would thank you very much for this talk. I know I've finished early, but it's like the other side of the.
Thank you very much, Dr. Taysir. I hope to catch a couple of minutes at the end of this session for questions, but our next speaker is Dr. Mona Bakr. Dr. Mona Bakr gained her PhD in 2002 from Georgia Tech in Atlanta, and currently she is associate professor at the National Institute of Laser Enhanced Science, Cairo University. Just to save time, as I did for the other, she is working on constructing novel nanomaterials for solar cells application, photoelectronic devices, biomedical imaging, cancer therapy, nano catalysis, and water treatment. Please join me in receiving Dr. Mona.
Good morning, everybody. I'm so glad to be here. And today, my talk about nanotechnology and how to localize nanotechnology in Egypt. And we've been working so hard for the last 10 years, of course. First of all, I would like to thank all of these people who really contribute in my career. First of all, Dr. Mustafa Said, he's my graduate advisor in his group since I've been with Mustafa for 20 years. And I learned, he is the one who's introducing me to nanotechnology and laser. And Dr. Ahmed Bahgat. Dr. Ahmed Bahgat, he's a businessman, and he has 2 PhD degrees. He started his career in R&D, and he's the one, without his support, memorial and financial, I will not be in Egypt. So he invested in me and in my group more than 20 million Egyptian pound to establish nanotechnology group to try to link between academia and industry and develop new material to serve Egyptian industry. And of course, our collaborator with whom we are working very intensive, Dr. Sami Shal, VCU in University of Virginia at US, Arturo from Spain, Stas from Texas, and Jackie Yan from IBN Singapore. This is the research group where I graduated in 2002 with my PhD. Back to nanoscience, you know, the dream of any nanoscientist, like the beautiful talk we heard in this morning from Dr. Hussein, how to really links between the structure and the properties, how to design material, manipulate the chemical structure of the material to reach whatever property I want. So our group is really very good in this. We are really can control the size and shape of nanomaterial, different material, plasmonic nanoparticle, carbon nanomaterial, graphene, semiconductor quantum dot, and I have more than 25 students are really good in making beautiful nanomaterial. Not only making material, we have to understand the relationship between the structure and the material itself and the properties and how to functionalize this material, play with the surface to make it fit any application. So gold nanoparticle could be used in cancer therapy, could be used in diagnosis, and also could be used as beautiful plasmonic material to enhance electronic devices like solar cell and plasmonic couple electronics with the photonics, so many catalysis, so many fields, the same material. You know, recently we prepare hydroxyapatite. It's non-dental material. This hydroxyapatite can be used as a feed additive, can be used in anti-corrosion bending, can be used in bone crafting and biomedical material, can be used in water treatment. The same material has many different uses. It depends how to really play with the surface and how to engineer this material and design it to fit the application. And we have to work side by side. I am a material scientist, I am chemist originally, understand a lot of physics, you know, but we have to work side by side with the medical doctor, with the biologist, with the pharmacist, with the engineer to develop this material to fit the final application we want, okay? So, of course, nanomaterial, all types of application from catalysis, energy conversion, energy storage, ultra supercapacitor, computers, sensing and biosensor, and we can, you know, to make all of this, there is a risk and there is a benefit. Any new material has risk and benefit. And we heard this beautiful talk about nanotoxicology. So whatever material we develop, we have to understand the environmental risk of this material and also the toxicity of this material in any type of application. Even the bent we have, if we touch it, what will happen? How it will interact with our skin. It will reach our blood and penetrate through our skin or not. We have to understand all of this. So we always have very good team of people who do the toxicology of this material for us and do different types of administration and study the effect or the hazard of this material and tell me what is the safe area I can work within. Of course, also like we heard this morning, gold nanoparticle has different shape and different sizes, and we prepare these particles since 1997, and how to prepare gold nanorods and controls aspect ratio of the rods, and control all of these beautiful physical properties, electronic properties, and optical properties. Also quantum dot, beautiful physics in the quantum dot and quantum confinement, and really how the exciton form, and all of these properties related to the surface and exciton trapping, and how to really engineer the material to enhance the electron-hole combination, to make lasers out of it, to make light emitting out of it, diode, or to make it photocatalyst to absorb sun and convert it into electron hole pair and do photodegradation, or solar cell application, all of these beautiful processes. And we are really good in making beautiful material, whatever the material is. Metal, semiconductor, make bimetal together, nano alloy, controlling the size and shape of it, making nanorods, making tetrapods, making wire out of it, and we are good in this. Also, not only this, we make hybrid nanomaterial, mix carbon nanotube with the plasmonic and see the interaction and the interface between them, and how this affects the optical properties and the electronic properties and surface properties and so on. And of course, the wonderland of graphene discovered in 2004. Graphene discovered in 2004, and they got Nobel Prize for discovering graphene in 2010. Since 2010 till now, there is 35,000 publications on graphene, and there is 25,000 patents in graphene. Any material you put it with the graphene, it has totally different optical properties due to the coupling between the sp2 hybridization, the electron cloud over graphene, and whatever you put on it. You put semiconductor, you put metal nanoparticle, you put plasmonic, all beautiful properties start to come, new avenue, new optical properties, new material, can fit any type of application, start from catalysis until anti-corrosive paint, until, very strong material, the most strongest material. Also, the electron transport in graphene 150 times better than silicon. So all people talking about graphene valley now. Also, the thermal conductivity. If we talk about copper, the most thermal conductor, 34. Graphene is 5,700. So thermal conductivity is the best. Photo absorption, absorb in the near IR light, So all the graphene properties is really amazing. So we are working intensive in graphene now and putting plasmonic into graphene, and we have a patent because this is very, very high efficient material to absorb light and convert it into heat. And we made solar concentrator out of this graphene plasmonic nanocomposite. And we use also graphene for biosensor, we use graphene for catalytic application. And actually, this is general what we can do, but actually this businessman, Dr. Bahgat, shook hands with me 9 years ago and really established nanotech, which has a lot of facilities like electron microscope, like very nice chemistry lab, very well equipped. And when we start to move from lab scale to production scale, we shake hand also with other industries like paint industry, like plastic industry, like other industry who suffer from environmental problem like water and stuff. And we take sample from them and we find a solution for them in the lab. And we have to produce this in a large scale, in ton scale. So we built our factory, which we can produce tons of nanomaterial. So we are the first people in Egypt succeed to move science from lab scale to production scales. And our lab has very, very nice facilities. Also, we have 5 tons or 5 meter square mixers and reactors and filter press and oven to dry and all of this. And we develop material for smart painting, metallization of plastic, for textile industry, silver nanoparticle, antimicrobial textile, softener for textile. We also produce so many material for agriculture and feed additive. And some of the application we are doing, it's biomedical application, like cosmetics, like also pharmaceutics, drug carrier and stuff, but this is long term because of the regulation in Egypt. We develop also material for water purification and for solar cell application and sun harvesting and stuff. 6 months ago, the government and Cairo University hired me to be the director of the Egypt Nanotechnology Center, because private sector can't do it alone. We have to really, localize this nanotechnology and work with Ministry of Health, Ministry of Agriculture, Ministry of Industry, Ministry of Environment to make a factory. We have to have the legalization from the environmental authorities to make you allowed to produce this material, and what is the waste, how they can deal with the waste. When you transfer nano material from place to the other, what happen if accident happen and this truck fell down and all of this environment be polluted with the nanomaterial. So we have to work with them to make the legalization and the industrialization of the nanotechnology to industrialize this and localize it in Egypt. So we are working hard in this, and through my position with Cairo University, IBM Center made this nanotechnology center just for the sake of the nano-electronic devices, solar cell devices, and system, solar concentrator for water desalination. When I came, we restructured the whole thing, and I told them, nanotechnology is a technology for the poor, and our country, we will not be able to compete with IBM or Intel. I want to make a material to help my society, to help my industry. What's my industry need? Textile, construction material, paper making, petrochemical industry, agrochemicals, pharmaceutical. So we made half of the institute now working to develop material and bring projects from industry to really help this industry. Material made with foreign know-how, and we imported with a lot of dollars. We try to reproduce it with Egyptian know-how and make it through our institute now. Also, we are trying to launch this with all Egyptian authorities in Egypt, like Ministry of Education, Ministry of Industry and Trades, Ministry of Defense, Ministry of Environment, also private sector, and we need the media and NGO to launch this with us, okay? So the first project actually I got through this beautiful collaboration between all of these alliance, gathering together the Ministry of Industry, the Ministry of Agriculture, the Ministry of Environment, is full utilization of rice husk and straw to produce nanomaterial. And actually, my group in the lab, so many people ask me to deliver nanosilica for them. And instead of making nanosilica from very, very expensive chemicals, like Taus and all of these reagents, no, we will make it from rice husk and straw. And actually, rice husk and straw, it's a huge problem in Egypt. Egypt produces more than 5 million tons every year rice straw, and about 1 million tons rice husk. And all of this huge environmental problem because they can't deal with it. Ministry of Environment can't deal with all of this amount, and they just burn it and make all of this pollution, and the black cloud cover all Cairo and all Delta area, okay? So my group took this rice husk and rice straw and extract beautiful chemicals out of it, cellulose, which can be used as a pulp for textile industry and paper making. We have our know-how to change the cellulose to nanocellulose, and nanocellulose has so many different applications I will talk about. And we can produce nanosilica with 99.9% purity, and more than 20% of the rice husk give pure nanosilica. So this has huge environmental impact. And you see, this is the structure of the rice husk, This is the structure of the rice husk. It has lignin, it has hemicellulose, it has cellulose, and little tiny nanosilica particle encapsulated inside to reinforcing the rice husk. Okay, so we have our chemical method of treatment to extract all of these chemicals. I can extract 15 chemical compounds. All of them have huge economic impact and can be raw material for many different industries from rice husk. Okay, so rice husk and straw, it has cellulose, you know, it has hemicellulose, it has lignin, and it has an ash, and this ash is mainly silica nanoparticle, pure silica nanoparticle, and you can see in this table the ratio of these chemical compound in the husk and in the straw. Also, this lignin can be converted efficiently. Okay, can be converted efficiently into phenol, and phenol is really has huge application in pharmaceutical industry and considered to be also biopolymer and stuff. So these are the application of the nanocellulose in different industries, you know, and this is application of nanosilica also in all different industries. And we make this alliance who really link between industry and academia to make, building in this research center of building. These people really take our nanocellulose and take our silica and make high performance concrete, make anti-corrosion bend. People from petroleum industry in the Research Institute of Petroleum take this nanosilica as a catalyst support. People from plastic industry take this nanosilica to reinforce and increase the mechanical properties of plastic. So we make this alliance, 15 institute and government sectors and private sector to collaborate with us and to solve this environmental problem. And we got fund from National Academy of Science of Egypt. 10 million Egyptian pound just launched this alliance, and we start already to make pilot, take 1 ton of the rice husk or rice straw and convert it to this really high value material. The same thing we did with Africa. The same problem with the rice husk, they have problem in pineapple. All of these tropical countries, Ethiopia, Malawi, Kenya, Nigeria, they have huge amount of pineapple. And all, of course, pineapple waste, the same thing. It's huge hazard for them, environmental hazard for them. So we can take this pineapple, the corn and the peel, and also extract this wonderful material out of it. I will stop here, and thanks so much.
Thank you very much, Dr. Mona. Dr. Mona actually rang the bell about the need to establish a regulatory agency in order to regulate the use of nanomaterials in this country as well, because it took almost 8 years to finish or to come close to finishing the risk assessment evaluation of silver nanoparticles to grant the approval of the United States Environmental Protection Agency as an agent in water treatment filters. So we must think of having a regulatory agency here in Egypt also to look after the risk and benefits or this environmental and health impact of nano products. We have a couple of minutes. We are actually 5 minutes late for the session, but we can have 2 minutes for any question and answer. Please, to the microphone. Your name and the question in brief. And
Dr. Mona Zaid, Associate Professor in Agriculture, Faculty of Agriculture in Shams University. For Dr. Mona, thank you for your lecture. It was very wonderful for me. May I ask you how you use nanocellulose and nanosilica in agriculture? The application?
Sure.
Use it in what?
Okay, nanosilica is known as pesticide, because pesticide and pesticide carrier. If you check the literature, of course, I am not agriculture, I am a material person. But we work with so many people in agriculture, and they use nanosilica as a pesticide, because it really can attack the skin of the insect, and it penetrates through, and it really cause problem, and it's a very efficient pesticide. Also pesticide carrier. All agrochemicals, growth hormone, pesticide, fertilizer, if you load it in nanocellulose, you increase the efficacy and you decrease the toxicity, and of course increasing the efficiency means less fertilizer use, means less pesticide use, means more efficient, of course, and it has economic value. But this is, we need to develop. We have group from Kafr Sheikh University, from Asyut University, and also agriculture company working together with me in this aspect, with also Cairo University, University of Agriculture. So we are working in this to stabilize it and optimize the condition.
Okay, another question, please. Go ahead. Dr. Samia, to the microphone, please, Dr. Samia. Dr. Salah, to the microphone.
I am Talwa Abdullah, Senior Researcher in Agriculture Research Center. I am asking Dr. Mona if we can apply the same approach of nanotechnology on botanical extracts or oils to be used as pesticides or not.
So many people use the oil and make the nanoform to increase also the efficacy of it, and I know a couple people, they ask me to convert oil like sesame oil and all of these things to nanoform and use it as a pesticide. But I am not the expert. I am just material designer. Okay?
So you are not a biologist in that sense. We can take the last question from here.
Dr. Amr from City of Scientific Research and Technological Application. My question also for Dr. Mona, because your talk was very interesting. You mentioned in your talk about using nanomaterials to make some animal feed additives.
Yes.
And I'm really interested for this point because I'm a nutritionist. Especially I'm working on animal nutrition, ruminants. So okay. Did you produce anything about...
Actually, we have a patent and we have product to launch in the next few months. And we did experimental in Agricultural University in Egypt, and we did in huge farms for poultry feed additives, and it will be launched very soon. It's nano calcium phosphate compound, and the efficacy increase 100 times more. And our product called NanoFus, and we are in the last stage of the regulation with the Ministry of Agriculture now to make it proof, and we will launch it. We are making some research also using nanoselenium, nanozinc and stuff, but still in the research.
It's okay. Thank you.
Thank you, and thank you, Dr. Mona, for your response for those questions. But let me have it straight, actually, because I have a debate, a long debate that took almost about 20 years now since they started to quote rice straw burning as a cause for the black cloud in Cairo. This is fake. This is not correct. We can have one of our students, maybe at the third grades, fourth grades, to tell us that the wind in Egypt comes from the west, from the northwestern side. So to get the clouds from burning the rice straw into Cairo, so we must have all of our rice produced and burned in Fayoum, or maybe in 6 October or so. And so the cause of the cloud is actually a photo-oxidative smog. Has nothing, well, might add to it if you have burning agricultural waste inside Cairo, helping that, but cars and there's states running in Cairo have the cause for that problem in that part of our country. Thank you very much for having this session. And I thank all the speakers here, Dr. Hussein, Dr. Jenkins, Dr. Taysir, and Dr. Mona for the lovely talks that we have with them. We have to come back here at 11 o'clock. You have coffee break, short coffee break, but we have to assure that we are coming here at 11 o'clock. Thank you.